Smoky Smokes. In this video on the nervous system, I'm going to be talking to you about the formation of a generator potential at the receptor, and then the formation and propagation of action potentials along the axon of a neuron until it reaches the axon terminal, at which point it will trigger the release of neurotransmitters under certain circumstances, but that is for another video. So let's start thinking about what a generator potential is. Essentially, a generator potential is just the very first action potential. And this term action potential is the term we use to describe this electrical impulse that is transmitted along an axon. In order to understand how this electrical impulse is transmitted along an axon, the first thing that we must understand is that our nerve cells have charge. They have something called a resting potential. This means that when nothing is happening, when they're not receiving or transmitting any sort of information, they already have a charge. The resting potential for our neurons is around minus 65 millivolts. We call this the potential difference, or the PD. You can just think of this as exactly the same thing as voltage. So let's set the scene. When we're drawing our neuron, we really need to be able to visualize it in our head. Obviously, we can draw it in 2 dimensions on a page, but that's not really that helpful. It's really important that you try and see it as a 3-dimensional thing so that it makes more sense to you. So when we're drawing it, we can try and draw it in 2 ways. So let's just jot down a generalized structure of a sensory neuron to remind ourselves. And let's make sure we can think about and visualize the axon both longitudinally and transversely. So if we think about it transversely, that would be if we were cutting it along here and looking down, and longitudinally is obviously this way. So if we imagine here is a longitudinal section of our axon, and here is a transverse section. Embedded into the membrane are sodium-potassium pumps. These sodium-potassium pumps pump out 3 sodium ions at the same time that they pump in 2 potassium ions. This is a pump. That means that it is active transport. So the sodium is being actively transported out and the potassium is being actively transported in. So it requires ATP, and the sodium-potassium pump is attached to an ATPase enzyme. I'm just going to introduce you to a couple of extra terms here. They're not super important, but they might be worth knowing. So transmembrane proteins that move molecules in opposite directions are called antiport, and transmembrane proteins that move 2 different molecules are called co-transporters, so you will often see these referred to as co-transporters. Alongside this active transport that's occurring, there's also facilitated diffusion. So we know that these charged ions cannot move directly through the phospholipid bilayer because they can't move through the fatty layer in the middle, and so they need to move through protein channels. There are many, many open potassium channels embedded in the membrane that allow potassium to readily diffuse out of the axon into the tissue fluid. There are also some sodium channels, but they are far, far fewer. That means that sodium diffuses much, much slower into the axon than potassium diffuses out. So, if we look at the overall effect on charge, we are actively transporting more positive ions out than are being actively transported in, plus we are allowing more positive ions to diffuse out in the form of potassium than we are allowing to diffuse in in the form of sodium ions. And so overall, it is more positive outside than it is inside. There is an overall excess of positive ions on the outside. This is why we say that at rest there is a relative potential difference inside the axon of minus 65 millivolts. This can also be expressed in graphical form. So if we look at a graph of potential difference and time, we can see that when nothing is happening, the axon is at resting potential. It sits at minus 65 millivolts. It's obtained, 1, by the sodium-potassium pump, and 2, by the fact that the membrane is relatively more permeable to potassium in comparison to sodium. Stimulus. Something touches the end of our receptor. This stimulus causes a physical pressure on the membrane. This physical pressure causes the opening of stretch-mediated sodium ion channels. So let's look at what would happen. The stretch-mediated sodium ion channels open. Sodium ions move down their concentration gradient. They flood into the cell. When they move into the cell, this increases the potential difference. Now, the potential difference is higher than minus 65 millivolts because there are more positive ions in the inside. If enough stretch-mediated sodium ion channels open, that enough sodium floods into the cell, that it causes the potential difference to rise to minus 40 millivolts. That is known as the threshold value. And at the threshold value, voltage-gated sodium ion channels will open. More sodium will flood into the cell. Sodium will keep flooding into the cell until it reaches plus 40 millivolts. When it reaches plus 40 millivolts, the membrane has depolarized. At plus 40 millivolts, voltage-gated sodium ion channels close and voltage-gated potassium ion channels open. What effect will this have? Well, now the potassium is going to flood out down its concentration gradient. As the potassium floods out, it will lower the potential difference. We say that it is repolarizing. However, what we see happening on this graph is it doesn't just reach minus 65 and then plateau again. What we see is it drops below minus 65. When it drops below minus 65, we say that it is hyperpolarized. It remains in a state of hyperpolarization for a very short period of time, and then it returns to the resting potential. But how does it do this? Once it overshoots the resting potential, once it enters a depolarized state, all excess gates close. So the stretch-mediated gates close, and the voltage gates close. All the gates and the sodium-potassium pump that were open at the very beginning during resting potential, they just stay exactly as they are, but all excess gates close. So now there's not all this extra movement of sodium and potassium ions, and the membrane returns to resting potential. We call this period of time where the membrane is hyperpolarized, so less than minus 65 millivolts, the refractory period. So what we have talked through here is the formation of one action potential at one point on the membrane. So just here, the very first action potential that is produced by a receptor is known as the generator potential. This is then propagated down the axon towards the axon terminal. So let's have a think about how it is propagated along. So let's go back to the basics. Here's our axon. The direction of travel is in this direction, and we know that at this end we have just generated our generator potential, and what that means is sodium ions have flooded in. Once these sodium ions have flooded in, they are going to diffuse within the axon. As they diffuse down the axon, if they raise the voltage of the next section of the axon enough so that it reaches minus 40 millivolts, it will trigger the opening of voltage-gated sodium ion channels in the next part of the axon. When these sodium ion channels open, that will cause sodium ions to flood in. This will depolarize the membrane until it reaches plus 40 millivolts. At plus 40 millivolts, we say that the membrane is depolarized. The voltage-gated sodium ion channels close, and the voltage-gated potassium ion channels open. Potassium floods out. The membrane becomes repolarized. It becomes hyperpolarized, and it enters a refractory state. After a very short period of time, it will reach resting potential again. This process of diffusion of the sodium ions along the inside of the axon causes what we can sort of imagine as a kind of Mexican wave of action potentials travelling along the axon towards the axon terminal. So one action potential causes the entry of sodium ions. The diffusion of those sodium ions within the axon triggers the next action potential, which triggers the next action potential, and so on. So it's like a knock-on effect. One action potential causes the next. This creates a wave of depolarization. The formation of an action potential in one section of the membrane causes the action potential in the next section of the membrane to be formed. So we can sort of think of it like one action potential triggers the next action potential, triggers the next action potential, and so on. But when the next action potential is being formed, so when this one is being formed, this one is in its refractory period and cannot form an action potential. That was just depolarized is now in its refractory period, so cannot be depolarized. So why does the action potential only travel in one direction? Well, let's just think about it. In order for you to become depolarized, you need to reach the threshold value. When these sodium ions are diffusing in all directions, when they're diffusing forward, they're entering an area which is at about minus 65 millivolts. And so they can raise it up to the threshold value of minus 40, and that can trigger an action potential. But when they travel in the other direction, that part of the membrane is in its refractory period. It is below minus 65 millivolts, maybe minus 80 millivolts. It would require more sodium ions to enter than actually do for it to reach the threshold value. This is why the refractory period is so important, because it ensures unidirectional flow. It prevents the action potential from travelling backwards. Because the membrane is temporarily hyperpolarized, it cannot reach the threshold value. So the refractory period can be split into two phases. The first is the absolute refractory period, during which time it's absolutely impossible to create a new action potential. And the second is the relative refractory period, where technically you could generate a new action potential, but you would have to have a very, very high intensity stimulus in order to generate that action potential. So refractory periods serve two purposes. One, to ensure unidirectional flow so that the action potentials are only travelling in one direction. And two, they ensure that the action potentials are discrete. That means that only one action potential can occur at one point on the membrane at one time. They don't overlap. Action potentials work on an all or nothing principle. This means that you either create an action potential or you don't. There's no such thing as a small action potential. There's no such thing as a large action potential. An action potential just is what it is. The values don't change, so you never go above plus 40, because the point at which you reach plus 40, it causes something to happen that causes it to repolarize. So in this video, we have looked at how an action potential is formed, so the series of events that occur within the membrane to trigger the formation of an action potential. We have looked at how that affects the voltage in graphical form of the neuron. And we have also looked at how action potentials are propagated along the neuron via the diffusion of sodium ions along. So as the sodium ions diffuse along, they increase the potential difference. And if they increase it enough to get to the threshold value, minus 40 millivolts, that will trigger the opening of voltage-gated sodium ion channels in the next part of the membrane and trigger the next action potential. We've also talked about the importance of the refractory period, how it prevents action potentials from going backward, and how it causes action potentials to be discrete. We've also talked about how action potentials are all or nothing. They either happen or they don't. We've covered a lot of information in this video. You are going to want to take a bit of time to write out the process, draw the diagrams, and test yourself to check you understand what's going on. But well done for now.